Hi everybody, welcome back to this short course of Bioinorganic chemistry. We have been discussing our life with dioxygen. Let us now go back to see what I have discussed in my previous lectures. I have discussed how dioxygen is reduced to water by the use of 4 protons and 4 electrons in cytochrome C oxidase, which is the terminal member of the respiratory chains. And you see that the important reactions is O2 converted 2 molecule of H2O, and you see that 4 proton and 4 electron have to be added all at a time with an E0 value of 0.815 volt. The dioxygen getting reduced in cytochrome C oxidase as I have just shown, and the reduction center is shown over here, as one can see that this iron to heme centers are this heme A3 and copper B, and heme A3 is 5 coordinated, copper B is 3 coordinated, and the structure is shown here, where dioxygen comes and binds perfectly between these two metal center. The X structure is displayed over here. You can see yourself how dioxygen fits perfectly between iron and copper centers. Now, this converting oxygen to water is very important reaction. This reaction represents the major source of energy when coupled with the oxidation of organic food. And in the respiration, as I have discussed, that the glucose and 6 molecule of dioxygen leads to 6 molecule of carbon dioxide, 6 molecule of water, and releases huge amount of energy which we use in our day to day activity. If the reduction takes place in a stepwise fashion, then dioxin would produce superoxide, peroxide and hydroxyl radical. I have discussed in details that these are called reactive oxygen species, and these reactive oxygen species are responsible for various age related diseases in our body. And we have discussed these in details in my previous lectures. Now, this superoxides, which is extremely poisonous in our body, converted to dioxygen and also the peroxides. So, one molecule of superoxide oxidized to oxygen, one molecule of superoxide getting reduced to peroxides, and as you all know, this reaction is a spontaneous reaction. It is called dismutation or disproportionation reaction. And although this is a spontaneous process, nature designed an enzyme called superoxide dismutase, which indeed immediately decompose these superoxides with a K catalyst value of 2 into 10 to the power 9 mole inverse second inverse, and destroy these superoxides to peroxides and dioxygen. Indeed, this peroxide which is produced in this reaction is also a highly poisonous, and nature has designed catalase and peroxidase in such a way that these peroxides convert to water and dioxygen. I will discuss these things in details. Now, this peroxides is extremely harmful, and it is an undesired intermediate in the dismutation reaction. Now, nature need to develop a defense mechanism to protect from peroxides. Indeed, nature has utilized this undesired intermediate to a extremely useful products using various enzymes such as catalase, peroxidase, chloroperoxidase, lignin peroxidase and cytochrome C peroxidase. Today I will be talking about catalase and peroxidase in details. Now, as one can see, this catalase and peroxidase going through a common intermediate iron 4 oxocation radical over here. Indeed, hydrogen peroxides oxidize this heme center, iron 3 heme center to iron 4, and which is a highly reactive intermediate, and in case of catalase, this hydrogen peroxide is a substrate which converts to oxygen and water, whereas in peroxidase the substrate is getting oxidized to convert something very useful oxidized product and converts back to iron 3 heme center, which again getting oxidized to iron 4 and goes back to iron 3. So, as one can see that iron 4 is a common intermediate for both catalase and peroxidase. Now, let us discuss one by one in great details. Now, catalase is an enzyme found in nearly all living organisms exposed to oxygen, and it protects cell from oxidative damage. Catalase also catalyze the decomposition of hydrogen peroxide to water and oxygen. As one can see, hydrogen peroxide is converting to water and also dioxygen, and the enzyme catalase which is responsible for this transformation is shown here. This is the structure of the enzyme at the resting state, and you see this iron is sitting inside the porphyrin ring, and there is 4 iron nitrogen bond, and fifth position is, as one can see, that this is the tyrosinate, and the sixth position coordinated with a water molecule, and iron is in iron 3 oxidation state. So, this is basically the structure at the resting state of the enzyme. In presence of this catalyst, this transformation becomes highly efficient, and K catalyst is 10 to the power 7 mole inverse second inverse. And X-ray structure of this catalase enzyme is shown over here. As one can see that heme center is sitting at the center in red in color, and you see that a huge amount of protein which are indeed wrapping the molecule. And if I remove this protein chain, what you can see that you can see only heme center is shown over here, and this is iron is sitting on the plane of the porphyrin ring, and a water molecule is weakly ligated at the 6th position. So, this is what is the resting state structure of the catalase enzyme. Let us look at the catalytic cycle of this enzyme. Now, this is what is iron 3 center at the resting state, and hydrogen peroxides oxidize this iron 3 center to iron 4 oxo cation radical. It is a 2 electron oxidation, as you can see that iron 3 converts to iron 4, and porphyrin ring also getting oxidized to porphyrin pi cation radical. Now, this 2 electron oxidized species is highly reactive, and it takes another molecule of hydrogen peroxides as a substrate, converts to water and dioxygen and goes back to the resting state, which again getting oxidized by using hydrogen peroxide to iron 4 oxo cation radical. So, this is what is happening in the catalytic cycle. It has been established by use of leveled hydrogen peroxides, where oxygen is leveled, that the dioxygen formed is derived from hydrogen peroxides. That means that O bond cleavage does not occur during this catalysis process. So, hydrogen peroxide, this O bond remains intact. Now, let us talk about another enzyme called peroxidase. Peroxidase catalyzes the oxidation of a wide varieties of organic and inorganic substrates by hydrogen peroxides. Overall reaction, as one can see, that this is what is the substrate AH2, and in presence of hydrogen peroxides and in presence of peroxidase enzyme converts to water, while substrate is getting oxidized to A. So, in presence of this peroxidase enzyme, the substrate is getting oxidized to A, while hydrogen peroxides, which is extremely poisonous, converts to water molecule. So, the substrates are generally organic molecules and can be fatty acids, phenols, amines and other species. Now, let us look at the structure of peroxidase enzyme at the resting state, as you have seen in case of catalase. Here also this iron is sitting at the center of the porphyrin ring, and as you can see there are 4 iron nitrogen bond, and fifth position is an histidine residue, while sixth position is occupied by a water molecule which is weakly coordinated to iron. Now, there are several peroxidase enzymes which are well characterized now, and some of them are shown over here: horseradish peroxidase, cytochrome C peroxidase, glutathione peroxidase, myeloperoxidase and ascorbate peroxidase. These are all well known peroxidase in the literature today. Now, horseradish peroxidase is well characterized, and X-ray structure is shown over here. As one can see this heme center, just like we have seen in case of catalase, sitting at the center, and the huge protein chains which are surrounding that heme centers. Indeed, these protein chains actually controls the specificity of the reaction, like the type of substrate that would be allowed to come in close to the heme center. I will discuss this in details, and if I remove this protein, you see that this is what is the heme centers, and this is iron which are sitting on the plane of the porphyrin ring, and this is histidine moiety, and the sixth position, as I have said, that it is weakly coordinated to a water molecule. And this is another X-ray structure of ascorbate peroxidase with ascorbic acid as a substrate. You can see yourself, this heme center is sitting at the center, and you also can see the ascorbic acid is also placed within this protein chain. And if I remove this protein, what you can see that this is the ascorbic acid. This ascorbic acid is used as a substrate, and this is the reaction with ascorbic acid. You see that this is the ascorbic acid, and in presence of peroxidase enzyme and with the help of hydrogen peroxides, this ascorbic acid actually oxidized to dehydroacetic ascorbic acid, as one can see that this 2 OH group is getting oxidized to the CO groups, and this is the ascorbate peroxidase which is responsible for this transformation of ascorbic acid to dehydroascorbic acid. Now, let us look at the catalytic cycle into more details. As one can see that this is the resting state of this enzyme iron 3 heme centers, which oxidized by hydrogen peroxides converts to iron 4 oxocation radical. This is 2 electron oxidation, where iron 3 is oxidized to iron 4, and this porphyrin ring also oxidized by 1 electron to convert it a pi-cation radical. So, this is 2 electron oxidized species called compound 1, and this iron 4 oxocation radical is highly reactive intermediate and also very similar as to the intermediate observed in case of catalase enzyme also. Now, this compound 1 highly reactive oxidized species transfer 1 electron to the AH2 substrate, and it forms AH radical and converts to iron 4 oxo species, which is also highly reactive. It is known as compound 2. Now, this reactive iron 4 oxo takes another molecule of substrate AH2 and converts to this AH radical, another oxidized product, and goes back to this iron 3 heme, which is present at the resting state. And this molecule again oxidized by hydrogen peroxides to form compound 1, and compound 1 convert to compound 2, and compound 2 goes back to the resting state. And during this catalytic cycle, as one can see, 2 molecules of H radical is being formed, and this H radical actually can convert to the oxidized species A, or A coupling can be there because these 2 radical can form a CC bond to one another and form a dimeric molecule like this. And so, now I will be talking about another very important enzyme called chloroperoxidase. This is the most versatile of the heme thiolate enzymes, catalyzing the chlorination of the activated CH bonds employing hydrogen peroxides and Cl minus at pH around 2.7, and the reaction is similar to peroxidase and catalase, as we have just discussed in our previous slides. So, overall reaction is shown here that AH plus X minus, X is the chloride. So, AH plus X minus plus H plus, and with the help of hydrogen peroxides, what is happening? So, AH converts to AX, and hydrogen peroxides the converts to water. So, while the substrate getting chlorinated is something very useful product in biology, hydrogen peroxide, which is extremely poisonous, it converts to water, very useful product for our environment. This is what is the structure of chloroperoxidase at the resting state. As one can see here also, a iron metal is sitting at the center of this porphyrin ring, and there are 4 iron nitrogen bond taking place, and 5th position is cysteine sulfur, you can see, and iron is iron 3 oxidation state. So, this is the structure of chloroperoxidase at the resting state, and one can see that this chloroperoxidase undergo lots of transformations in biology. Say one such example is shown over here, as you can see this molecule, this monochlorodimidone converts to 2,2-dichlorodimidone. You see that this chlorination is taking place at the 2 position, and this is highly stereospecific. X-ray structure of chloroperoxidase is shown over here. As you can see once again that this molecule, that heme center, is sitting at the center of huge protein chains. This protein chains indeed responsible for its specificity, and why it is so stereospecific. Now, once I remove this protein chain, you can see these heme centers. This is the iron, and cysteine sulfur is this, and this is the active site structure of the enzyme. Now, let us look at the catalytic cycle of chloroperoxidase. Now, as we have observed in case of peroxidase, the mechanism is very similar. So, what is happening? The resting state of the enzyme is shown over here, which is getting oxidized to iron 4 oxocation radical by hydrogen peroxides. It is 2 electron oxidized species, and as you all know that this intermediate is highly reactive. It takes a chloride ion and it forms that OCl bond, and where iron 4 converts to iron 3. Now, this OCl after one proton, and then it converts to OHCl, and iron 3 remains as iron 3, and this Cl actually chlorinate at this particular carbon and converts to 2,2-dichlorodimidone and goes back to the original species which are present in the resting state. So, this is the way this enzyme chlorinate stereospecifically the substrate. Now, we can learn lot of things from nature. So, as you have seen that in case of catalase, peroxidase and chloroperoxidase, the reactive intermediate like compound 1 is this iron 4 oxocation radical. Only difference was this axial ligand L, okay? And this is being formed using hydrogen peroxides. One can utilize this highly reactive intermediate, and substrate can convert to a variety of products, like sometimes it is an oxo transfer reaction can take place, sometimes it is an electron transfer reaction can take place, sometimes if it is like a chloroperoxidase, it can chlorinate at a particular position in a stereospecific manner. Now, we can make use of this process. We also can learn from such catalytic activation of hydrogen peroxide for a large variety of transformations that the enzymes have been doing forever. The process has immense potential for various practical utilizations in the laboratory and industry, such as cleanup applications for eliminating toxic and polluting substances and undesirable organisms. Now, what we learn from nature? This is the nature's design in catalase and peroxidase at the resting state. Now, the search for useful peroxidase catalyst has involved numerous groups for many decades, and important catalyzed processes have been developed in such field. The central mission of such green chemistry is to invent new processes that do not pollute our environment, improve the quality of our life and promote sustainability. One such commercially successful synthetic catalyst, Tamal, is shown over here, whose design is fully inspired by catalase and peroxidase enzymes present in our mother nature. This can replace chlorine technology with alternatives based on nature's oxidant, oxygen or hydrogen peroxide. Indeed, their active forms are also very similar. In enzyme, it is iron 4 oxo pi-cation radical, while in thermal catalyst it is iron 5 oxo species. As one can see, the design is fully inspired by catalase and peroxidase enzymes present in our mother nature. In conclusion, our mother nature has designed superoxide dismutase, catalase and peroxidase, which not only destroy those reactive oxygen species, but also converts them to something extremely useful for us. I have shown the enzymatic structure of catalase and peroxidase today and discuss about the mechanism of their actions in great details. I have also highlighted here how the beautiful design principle adopted by nature control all these processes in our life. In my next lecture, I will talk about the reactions of dioxygen with organic substrates catalyzed by oxygenase enzymes, in which oxygen atoms from dioxygen are incorporated into the final oxidized products. Indeed, dioxygen gets activated first and then inserted into the substrate, which we would be talking in great details in my next lecture. Thank you.